This is screen capture video 12.2, the second video in the series, and in the first video I left off talking about the structure of DNA. This is sometimes referred to as the ribbon model, the computer model over on the far right, and then in the middle here is just a partial structure, just taking a look at a few of these and pulling them over. Also a reminder, A always pairs with T and A pair with each other, and C and G always pair with each other. That's a given. That's just what they do unless there's a problem such as mutation. And if they do mispair, sometimes that does cause mutations. But A and T always pair with each other, and C and G always pair with each other. Complementary base pairing. Chargaff was working in the 40s and 50s, and Erwin Chargaff, and he understood that the percentage of adenine always equaled the percentage of thymine, and it would be within 1% in his experimental testing, and that was within the range of error that was acceptable. So for all practical purposes they were equal, and cytosine and guanine were always found in equal amounts. And probably I'll try to put some kind of a problem on the test where I give you one of these and you'll be able to calculate the percentage of any other. Keep in mind that adenine and thymine always equal each other. Cytosine and guanine always equal each other. So I don't have it typed out here, but if the amount of adenine and thymine were to equal 40%, both of those together, each one would be 20%. If adenine and thymine equal 40% of all the DNA, that means the cytosine and guanine would equal 60%. Half of 60 would be 30, so cytosine would be 30, guanine would be 30. There are different ways of looking at that, but I may very well ask a question on the test about that. I'll try to put one together for you. This is a picture of Erwin Chargaff. He's died just in the last, he died in 2002. Lived a fairly long life, lived into his 90s. But he was very instrumental in helping to understand the DNA molecule. The three people that actually won a Nobel Prize for discovering the structure of DNA were James Watson and Francis Crick and Maurice Wilkins. There was a fourth person who was involved in the process, but she died of ovarian cancer and complications of ovarian cancer before the Nobel Prize was awarded. But they were doing their work in 1952, 1953, and their discovery of the structure of DNA was in 1953. I believe it was April of 1953. And they were awarded the Nobel Prize, I believe, in 1960. And the reason it took several years, generally when there's work that's done, the scientific community wants time to review it, to do tests on it, to verify that it's correct, because Nobel Prizes have been awarded for things that are false, that aren't true. Maurice Wilkins and Rosalind Franklin, Rosie as she was known to her colleagues, some of them, X-rayed the DNA. This is back before they had electron microscopes, so they would crystallize DNA. It was a time-consuming process. But they would crystallize the DNA and then hit it with X-rays and look at the photographic plates. And at this time they found that there are two different types of DNA. One was alpha, or an A form of DNA, and this is an X-ray photograph of it. And notice dark banding here, across from each other, dark banding across from dark banding over here. And if you can almost imagine this, it's as if you're looking down a tunnel. And the way it was described by those who eventually discovered the structure of DNA, you're looking down the DNA molecule. I don't know how they did this, but you look down the DNA molecule and you can see these nitrogenous bases that are situated towards the middle of the molecule. The sugar phosphate backbone would be way out here. So this is an A form, and you can look at this on your own, and I've got links, I think. This is the B form. And the pattern of the B form is just a little bit different because of the way it crystallized. This is from Rosalind Franklin's X-ray photograph. Here's a picture of Francis Crick. He was a little bit older than James Watson, but he and Watson teamed up and worked together. This is James Watson. Crick was an Englishman. James Watson was American. He actually got money from the government to go over to Europe to study viruses, but he kind of got sidetracked and decided to study DNA because he thought that would be more exciting. Not the most ethical thing to do, but that's what he did. And this is Maurice Wilkins. And this is Rosalind Franklin. James Watson, in his book, The Double Helix, referred to her as Rosie. They didn't get along very well. Quite a bit of friction between them. And a number of people didn't get along with Rosie Franklin. Supposedly her mannerisms and her short answers and impatience with people didn't sit well with her colleagues. They felt that she talked down to them and didn't bode well for relationships. One of the things that we understand is that DNA comes from DNA. And DNA is made from DNA in a process called semi-conservative replication. And as I describe this process, I'll talk about the parent molecule, I'll talk about the daughter strands. And I'll probably mention the parent strands too, and then the daughter molecules. I'll try to explain those as I go through it. I do have a link for semi-conservative replication. And I'll show the link down below. Just as a reminder, if you do have the biology book by Campbell, you can see the structure of DNA. DNA is made up of adenine, cytosine, guanine, and thymine. When you see the thymine, that tips you off you're looking at DNA and not RNA. And showing it in a little bit more detail as you move to the right, you can see the phosphate, sugar, nitrogenous base groupings. Those are called nucleotides. And over here, they show a specific nucleotide, phosphate group, the sugar, and the nitrogenous base. It happens to be thymine. The nitrogenous base could be any one of the four. But this unit right here is called the nucleotide. And keep in mind that the DNA molecule is a double-stranded helix. Now, the work for determining that DNA is made from DNA in a semi-conservative process, this came from the work of Meselson and Stahl in the 1950s. 1957 is when the work was put forward. And I've got an illustration here that you can take a look at. What Meselson and Stahl's group did, they labeled the DNA with a radioisotope of nitrogen. They called it heavy nitrogen because it's a little bit heavier. It was nitrogen 15 versus nitrogen 14. They replicated DNA so that all their DNA that they're working with was nitrogen 15 DNA. And they put that into a centrifuge, had the cesium chloride in it, spin it down, and it would be at a certain level. And if you notice, it's a little bit subtle here, but notice it's lower in the tube because it's heavier. And they show the makeup being homogeneous of nitrogen 15. And when they looked at the banding pattern of it with the densitometric scans, it scanned very thick in this one region where it was heavier. Then they allowed the DNA to be replicated. Whatever process that happened, it was replicated. And they noticed that all the DNA was heterogeneous. It was made up of nitrogen 15 and nitrogen 14. And so when they spun it, it all spun in one pattern, in one layer, and it wasn't as low as the first one because it's not as heavy. And it showed up with a different scan and a different band. So that was interesting. That pointed to the parental DNA strands being incorporated into the daughter molecule. And as they continued to replicate it, went through a second cycle, then they saw something a little bit different emerge. Some of the DNA was all new, nitrogen-14, had no nitrogen-15 in it, and then some had nitrogen-15 in it. And this would give two banding patterns, a little bit more of the lighter DNA because more of this is being made, and you see the two peaks in the scan. And in the centrifuge tube, there are two different layers. In a third series, you're going to see more of the same, except the nitrogen-14 DNA is going to increase. You're going to get more and more of that. And the nitrogen-15, nitrogen-14 DNA, that heterogeneous mix, so to speak, it's just going to stay the same. And they saw two bands, the band or the layer of the nitrogen-14 growing. And you can see in the scans it's getting thicker and thicker. And the nitrogen-14, nitrogen-15, not as thick, not as dark, and no nitrogen-15, nitrogen-15. And here on the scan over here on the far right, you can see the nitrogen-14 peak is getting larger. And if we carried this out even further, it would get larger and larger and larger. And the peak for the nitrogen-15, nitrogen-14 pretty much stay the same. It would not increase in amount. So that helped to show the process of how DNA is replicated. So in semi-conservative replication, it means that the parental strands of the parental molecule serve as a template so that new nucleotides can come in and assemble, and the bases can pair with each other. And when these nucleotides come in close, they can be cemented together or bonded together. It's a better term. And so over a period of time, a new daughter strand forms along each parental strand. And eventually, the parental molecule completely separates, unwinds and separates, so that it no longer exists. What was part of the parental molecule now makes up half of the daughter molecule and half of the other daughter molecule. This is why it's called semi-conservative, semi-conserved. Whereas I did not put it in as an image, but it's possible that it could be completely conserved, where it just serves as a template, and you still have the parental molecule and then a whole new daughter molecule, which isn't shown here, but it would be as if these two daughter strands attached to each other. That doesn't happen. It's semi-conservative. This is showing a replication fork. What happens, the DNA unwinds at one end. This is a fairly specific process, and I'm not delving into it in great detail, but it involves 5' and 3' ends. One end of the DNA is 5' end, the other is 3' end. I'm not discussing that much. I'm not discussing a lot of the enzymes involved, the DNA polymerase, the ligase, the helicase. I'm not mentioning those. But at one end, the DNA molecule unwinds, it separates, and a daughter strand is formed in one direction along one of the strands. Along the other strand, it forms in pieces, and then those pieces are bonded together. It kind of goes in a different direction and a different method on the other strand. But in the end, you have two new daughter molecules. And in the next video, I'll talk about that a little bit more.